Last but not least, we have Jenelle Feather, who did her PhD with Josh McDermott and is wrapping up her postdoc work at Flatiron and starting her lab at CMU this fall. Jenelle. Oh, and she'll talk about exactly what Anya kind of ended on in her response to the last question.
Thanks so much. I'm really excited to be here today. So while I was here at MIT and McGovern, I worked with Josh McDermott. And so actually a lot of my background is in the auditory system, but generally I study both audio and visual perception. But today I'm actually going to be focusing in on some interesting ways that we can probe visual models of perception. And so that way, visual models are easy to look at the images, and we won't run into any audio problems. So broadly, my research focus is computational cognitive neuroscience, where I study how biological systems, such as humans, perceive and interact with the world. For instance, you can easily hear the pressure wave of your friend's voice as they tell you that they got a new pet. And when they show you a photo, you immediately know that they got a dog and that they didn't get a cat. The neural representations within our sensory systems mediate these perceptual tasks like word recognition or object classification. And as we've heard from many people today, these representations can flexibly change due to memory, learning, or constraints of our environment. So the intricate patterns of neural activity that underlie perception and cognition are really quite complex, but computational modeling provides one way that we can bridge the gap between observed brain representations and behavior. And recently, as we heard about a bit in the last talk by Anya, artificial neural networks have really emerged as some of our leading models in this space. So you've all heard about these AI systems and how they can perform naturalistic tasks like speech recognition, object classification, and many, many other human-like behaviors. And these artificial neural networks are really composed of sort of stacked building blocks that each building block itself is really a quite simple mathematical operation, but putting them all together allows us to do something much more complex. And so the real advancement in the last decade or so is that we can now stack many of these building blocks together, and we can train these models by giving them example data, and afterwards we have a computational model that can do many complex things. And so once they're optimized, because they can do these tasks oftentimes as well as humans, this has really led to widespread interest in seriously considering these models as candidate models of the human brain. But if we actually want to use these models to their full potential to do things like perhaps develop new algorithms for audio or visual prosthetics, or maybe to sort of propose some hypotheses for how the brain might also be implementing its own algorithms, we really need to understand what these systems are doing. We need to find their discrepancies with biology, and we need to make changes to them that help align the models with biological systems. And so one way that we've been approaching this problem is by designing stimuli that help us understand various aspects of the models. Essentially, what we want to do is really dig into these black boxes and try and understand various properties. So during my time at the McGovern Institute with Josh McDermott, we developed a framework for testing the invariances of these computational models, such as deep neural networks, by using the idea of model metamers. So model metamers are stimuli that are physically different, but that produce the same responses within a computational model. And thus, they capture the model's invariances. Now, for now, I'm just going to focus in on the classification decision of the model. So for instance, you might have an image of a bear here. You recognize this image very clearly. This is a bear. Now, this image, once again, is a bear. It's a bit different, but it's still a bear. And this image, again, this is still a bear. It's a different type of bear, but you are invariant to things like the sort of position, orientation, exactly the type of bear. And in fact, this is the type of thing that oftentimes we'll train these visual neural networks to do. We'll present them many images of bears and have all of them be labeled as a bear and treat it to label each of these images as a bear. So if we have a good model of human visual perception, we might expect that all images that are actually matched to the representation for this bear image will at least still look like a bear. And so what we did is we used the same tools that are used to train these deep neural networks, and we basically optimized the pixels of this visual input to find a new image that yields the same network activations as this image of a bear. And so if the models are good, we think that they might be because we use them every day, you might expect that, in fact, they have invariances that are similar to those of humans. But instead, this is the image that we get out. The model says that this image that really looks like noise is a bear. And in fact, these two images are model metamers for this particular visual artificial model. And so what that means is the model cannot tell these images apart, even though the one on the right is very clearly different and doesn't look natural at all. And so this was really a pervasive failure that we found within essentially all of the image models that we tested. It also was present in essentially all of the audio models that we tested, and we ran many behavioral experiments along these lines. But we didn't stop there. After all, we have to make these models better once we find problems with them. And so as part of this work, we also found ways that we could improve these models to better align their invariances with human perception. Basically, in this case, by highlighting these problems, we were able to then go and search for ways that we could improve them. In this particular example, this is using a technique which is called adversarial training. And for those of you who are sort of in the AI community, this is essentially reducing the adversarial examples in the model, which are also another one of these ways that we can sort of design stimuli to probe aspects of the model. Okay, so basically, model metamers were one way that we can design stimuli to tell us differences between computational models and human perception. But I also actually want to talk about another more recent project, which is part of my postdoc work, where we are also thinking about designing stimuli to tell us things about our models of perception. This work is really, I guess you could say, hot off the press. We're presenting it in 2 weeks at one of the big machine learning conferences. And it was done in collaboration with an amazing group of researchers at Flatiron Institute. And this includes my co-first author, David Lipschitz, on the left here, who just started his own lab at Baylor College of Medicine. So for this project, the idea is that we wanted to think about how we could actually design stimuli with the goal of distinguishing many different models simultaneously. Okay, so often when experimenters investigate similarity patterns of activity between models in biology, we might present many natural stimuli and look at how these stimuli cluster in the sort of model representation. Oftentimes, this comes down to categorical decisions, similar to what we talked about for the metamers work. So for instance, humans might say that animate objects like dogs and cats are close to each other, and inanimate objects like this tree and the car are far apart, but we might see that a model doesn't have this animate versus inanimate distinction. But it's possible that there's another model that actually does capture very similar patterns to those that were observed in human perception, where dogs and cats are represented very close to each other. And so many techniques have been previously proposed to look at various aspects of this sort of representational space. And oftentimes, this is called the representational geometry. And so essentially throughout this, the goal is to find models that have similar patterns to humans. And it's the case actually in the sort of intersection of neuroscience and artificial intelligence that we currently actually have a lot of models that seem to have very similar representations according to these measures. But even if the overall geometry seems very similar for the sampled set of stimuli, this is actually a very high dimensional space, and it's very complicated. And so there might be many other parts of the space where differences might be very pronounced. So how do we go about efficiently sampling aspects of this high dimensional representational space? Experiment time, as anyone who's run an experiment, we know it's very limited. We can only collect responses for so many images. And so how do we actually choose what images we should present? So what we did in this project is we really zoomed in on aspects of what we'll call the local geometry of these representations. So this is the geometry that's induced from very small perturbations to a base image. These types of changes in the representations are particularly relevant if you want to compare sensitivities to various distortions of the image. And so previously, there actually was some work looking at this local geometry for a single model, which suggested that what you should do if you wanted to compare one model with a human observer is in the model, you should find one direction that sort of maximally changes the representation and another direction that minimally changes the representation. And these are the optimal two directions that you would use in order to probe the human representational space. And so what you could do is find these directions in the model. So for instance, this model is very sensitive to perturbation A and not that sensitive to perturbation B. But then you basically present these same stimuli to humans and have humans see how sensitive they are. And so for this example, we actually see that the human sensitivity is flipped for these two distortions. Now, this is just a sort of two-dimensional example, but as some actual distortions for one of these deep neural network models, we can look at them. So this model in particular is more sensitive to distortion A, and the representation of this model really doesn't change for distortion B. And so now I'm going to show these distortions added to the image, and you can see where you might notice them. Here we have some chickadees. And so this is here just added by a very small amount. But as we go up, maybe you can start to see that these images are becoming distorted. I actually think that humans are a little bit more sensitive to distortion A here. But what you can actually tell from this is that these differences are really quite subtle. Here, we're just testing one model. And so say that actually we had 50 different models that we wanted to test with these very subtle differences between the distortions. This would be an absolutely terrible psychophysics experiment to run. So instead, in our recent work, what we did is we proposed a way to design stimuli that explicitly optimize for many models simultaneously. So instead of finding a direction that maximally changes one model and another direction that minimally changes it, we instead just find two directions that elicit a large amount of variability in the sensitivity across many different models. And then the idea is that we can run a human perception experiment to measure the sensitivity and eliminate models that are very clearly different from the observed human sensitivities. So here, we're actually considering around 50 different candidate models of human distortion detection, where we generated two distortions that push these models apart as far as possible. And you can start to see that there's actually noticeable differences between these two distortions, where the distortion on the top is sort of localized on the content of the image, where the one on the bottom is really localized in kind of this background information. And so once again, we can add these to the distortions, and even though they are quite subtle still, if we were running this experiment here, we would only need to be running this one example, at least to start, to test these models. And I don't have all human data today. We are currently in the process of running that, but just from sort of our own visualization, we would say that the human response for these sensitivities would be somewhere around here. And what's nice is that you could then eliminate the models that are really bad and generate new distortions for these images. So to conclude, I talked about these two methods that basically we have talked about in order to reveal some of these differences between the models and also use visual stimuli in order to help us with a model selection procedure. So hopefully I've convinced you that these networks are not just black boxes. And we can use sort of tried and true neuroscience methods using targeted stimuli to try and understand their representations. So with that, I would like to thank many people for this, all of my collaborators on the projects, and also thank McGovern Institute. I'd also like to thank Flatiron CCN, which is where I'm doing my postdoc. Soo-Yeon Chung is also one of my postdoc advisors. And as I've mentioned, I am starting at Carnegie Mellon in the fall. And so, yeah, happy to take any questions. Thank you.